Hello, grade 8 students. In this video, we're going to continue with our chapter digestion. And here we'll move to the food test, the second part of the food test, and the part 3 of digestion. So first, we talk about the Fehling test. We said that the Fehling test is done to identify the presence of reducing sugar. Mainly the reducing sugar, all of them they are reducing except for sucrose, which is a non-reducing sugar. So first test is the Fehling test, and the aim is to identify the presence of reducing sugar. What is the procedure? So first we put a food in a test tube, then we add equal amounts of Fehling A and Fehling B to the test tube. Okay? Then the obtained solution has a blue color. So first of all, we had equal drops of Fehling A and Fehling B, and the solution has blue color. It's called the Fehling solution. Then we heat gently the test tube over a Bunsen burner, which is the source of heat. The obtained results. If it's positive, we will have a brick red precipitate. Like brick red, it's the one here. It's the brick red precipitate. This indicates that we have reducing sugar. While if it's a negative result, so there is no change in the color, it remains blue in color like the Fehling solution 1. So this indicates the absence of reducing sugar. So you have to keep it in your mind. The Fehling test is done to identify the presence of reducing sugar. So if we have a brick red precipitate, this indicates the presence of reducing sugar. If we don't have brick red precipitate, this means that we don't have reducing sugar. Iodine test is done to identify the presence of starch. So here, as you see, the color of the test here of the solution, it's brown orange in color. Okay? When it's added to the starch, here they use cornstarch for example, it changed to be dark blue color. So whenever we have dark blue, this indicates the presence of starch. So the procedure, put a piece of food inside a small dish, then add few drops of iodine solution which has a brown orange color. If it's positive, it will give dark blue color like the case here. So this indicates the presence of starch. While if it's a negative result, so the solution remain brown orange in color. So this indicates the absence of starch. So iodine test is done to identify the presence of starch. Add a few drops of this solution on the piece of food to be tested. If it's a positive result, the solution the color will change into the dark blue. Okay, this indicates the presence of starch. While if it's a negative result, the solution will remain brown orange and there is no starch. Third test is the BioRed test. BioRed test is done to test the presence of proteins and slash or polypeptides. So either we have proteins and polypeptides, or we have proteins or polypeptides, one of them, or both of them. What is the procedure? Put a piece of food in a test tube, add equal volumes, meaning that equal drops, let's say, of copper sulfate, CuSO4, and sodium hydroxide, NaOH. So this mixture is called the BioRed solution, and it has blue color. So the BioRed is done to test the presence of proteins and or polypeptides. If it's a positive, we will have a violet color, a purple color. This means that we have proteins and / or polypeptides. While if it's a negative, there is no change in the color and it remains like the color of the BioRed solution, which is blue color.
Proteins can be detected by the BioRed test. To 2 cm cube of egg white solution, add BioRed solution dropwise. Shake after every drop is added. What color changes do you observe? If proteins are present, a violet color develops. If proteins are absent, the solution remains blue.
Moving now to the coagulation test. Coagulation test is done to identify the presence of proteins and only proteins. So we have to heat the food till boiling. If the food coagulates, if the food shrinks, this means that we have protein. If there is no coagulation, this is a negative result. This is the case of the coagulation. The food coagulates, it shrinks. For example, if you do a barbecue party, if you have a large piece of meat and you put it, for example, in the barbecue, what will happen? This piece of food will coagulate. This piece of meat will coagulate, will shrink. It becomes, for example, if it's like this, it will be smaller. It will be like this, smaller in size. It occurs in the burger sometime. Whenever you have a burger, for example, the burger, whenever you boil it and you put it over the Bunsen burner over heat, it will be smaller in size. For example, you see it like this, it will be like this. It will shrink. This is coagulation of the food. So whenever the food coagulates, this indicates the presence of proteins. Translucent spot test. This test is done to identify the presence of lipids. And this is the first test for lipids. We have another test. We're going to talk about it in the next slides. So, translucent spot test. It's done to identify the presence of lipids. What do we have to do? Rub a piece of food against a paper. Rub it, for example, a butter, for example, or nut, for example, containing lipid. Rub it with a paper and observe it under the sunlight. Mainly it occurs whenever you are eating manoushi and more specific za'atar. Okay, this manoushi that contains za'atar, it contains oil. So whenever you see this paper will become having spots like this. So if you observe it under the sunlight, it will be a translucent spot. It allows small amount of light to enter through it. So this is the translucent spot and it's a positive result, meaning that we have what? Lipids. While if there is no translucent spot, so there is no lipid and it's a negative result. The second test that's done to identify the presence of lipids is called the Sodan test, and not Sodan. Sodan test. Sodan test is done to identify the presence of lipids. It has a solution like the iodine solution. It has a Sodan test solution. If we add it and we have a pink color like the case here, so this is a positive result, meaning that we have lipids. While if it's a negative result, no pink color appears, meaning that there is no lipids. So this is the second test that is done to identify the lipids. The first one, it was the translucent spot test, and the second test, it was the sodium test. So till now, we have the different tests. We have the Fehling test for the reducing sugar. We have the iodine test for starch, BioRed test for proteins and slash or polypeptides. Also, we have the test which is the coagulation test for proteins, and the last two tests which are translucent spot and the sodium test for lipids. This is about the different food test for this session. Thank you guys.